So we left off the last video with talking about and looking at the video clip that showed crossing over. So make sure you have in your notes what synapsis and crossing over is, and then what we want to make sure you have as well is that you understand, like we talked about when the video was playing, that protein strands are holding the chromosomes together, they're stabilizing it, and then of course talking about that vocabulary term chiasmata, where the actual spot of crossing over is occurring. That's what chiasmata is. Now, the actual thing that does the whole process of switching the sequences of DNA, those are enzymes. Enzymes are responsible for actually detaching and reattaching the chromosome pieces. So we're going to move on to meiosis I. So meiosis I, metaphase I. This is where homologous chromosomes will line up in pairs down the middle of the cell. This is very different from mitosis. In mitosis, we lined up in a single file line. So this guy and this guy wouldn't be sitting next to each other, they'd be in line with each other. So this is already setting up, making that genetically unique offspring. We're already starting to see some genetic recombination. So when they line up in pairs like this, remember each chromosome has a version from mom and a version from dad. They don't care when they line up where they came from. So we could have both chromosomes from mom on this bottom side, and we could have both of dad's chromosomes up here, or we could have a mixture of them. It won't matter where they came from. They're just going to line up in pairs. This is called the law of independent assortment. And this random distribution of chromosomes lined up on either side of this metaphase plate is what gives us genetic diversity as well. This is another way for cells to get genetic diversity. So we've already seen two. The third type of genetic diversity that we can see is mutations that occur in DNA replication. So when we make copies of our DNA, sometimes errors occur. Those errors are called mutations. And those are the three ways we can get genetic diversity: crossing over, the law of independent assortment, and mutations. Then we have anaphase I. Anaphase is where the chromosomes get pulled apart to opposite ends of the cell. It looks very similar to what you would see in mitosis, except remember we have a full chromosome here. Instead of separating those chromatids, we're separating those homologous chromosomes. Now in telophase I, this is where the cells split and we get those two cells. The thing to remember here is that we have already established genetic diversity. These are already different than what you would see in mitosis. In mitosis, we would have one version of each chromosome in each daughter cell. But here, we have only the mom's version and only the dad's version for each chromosome. We do not have both versions in each cell. So just kind of comparing where we started and where we ended up. So a cell going into meiosis is what we call diploid. Diploid means that it has two versions of each chromosome. By the end of meiosis I, in telophase I, we're already establishing haploid cells. That means they only have one version of each chromosome. We've gone through DNA replication once before we even started meiosis I, and we're going to divide twice. We've done one division, so now we need to go through the second round of division. And that's where meiosis II comes in. Some very important things to consider when looking at meiosis II. The biggest difference between meiosis I or even mitosis is that we have two cells doing this now. Two cells are going to go through division. And we're again going to be seeing that development into haploid unique cells at the end. So prophase 2, we have the nuclear membrane breaks down. It's pretty standard prophase. Metaphase, again, here we see it looking very similar to mitosis with the big important difference that two cells are doing this now. We have our chromosome lining up in the middle, single file, but we have two cells where we've divided all of our versions between. Now they're going to get pulled apart, and we're going to end up with 4 haploid, genetically unique cells. We're not going to go through any more replications. This is what we wanted when we started out with meiosis. These are some terms to be familiar with. So we have gametes. These are sex cells. They contain half the number of chromosomes, so that means they're haploid. We have somatic cells, which are body cells. They contain a full set of chromosomes. We call them diploid. Haploid, remember, we can represent that as N. We can represent the total number of chromosomes in a diploid cell as 2N. So in humans, our diploid number, 2N, is 46 because that's the total number of chromosomes we have. And then we have the haploid number, which is 23, which you'll remember humans have 23 pairs of chromosomes. And if we take 23, multiply it by 2, we get our diploid number of 46. We, of course, have homologous chromosomes, synapses, chiasmata, crossing over, the law of independent assortment, and genetic recombination. Those are all things you should be familiar with as well. But we're not done. Just because we've gone through meiosis does not mean we have fully functioning gametes. We have to go through a process called gametogenesis. Gametogenesis is where we take the products of meiosis and make them functioning gametes. So here we have male gametogenesis, which is called spermatogenesis. And the process basically boils down to adding a tail. We call that tail a flagella. So each product of meiosis is going to be given a tail because we want our sperm to be motile. We want them to move. And we basically don't want them to have anything else. If they had anything like organelles or a ton of cytoplasm, they would be really slow. And sperm wants to be the fastest sperm it can be. It wants to get to the egg, put its chromosomes inside of it so that it is the one who passes on its traits to the next generation. So we want our sperm very fast, very streamlined. Because of that, sperm only really contribute DNA to the embryo. They don't really contribute anything else. Contrast that with female gametogenesis, which is called oogenesis. This is the process where we're creating the egg cell. The egg cell is going to be contributing all of the cytoplasm and all of the organelles to the future offspring. So meiosis occurs very differently in oogenesis. When we're creating eggs, the cells divide unevenly. So 90% of the cytoplasm is going to end up in one cell. So when we further go through meiosis, we end up with one cell that has all of the cytoplasm, all of the organelles available, and then we have these byproduct cells we call polar bodies. This egg cell is what's going